IIM 2015 for this outstanding meeting, which has been an inspiration to our young faculty for me also. And of course, the incredible venue in which we are having a great time. And thanks for having me here as a part of this endeavor. Like many of us, I don't know how much I can serve you as a mentor, but I am happy to share my experiences for the past 5 years, building up a lab from scratch to a functional one in a new institute like ISL Mohali. Like most of us, I am crazily passionate about science. And if I stop back and think that when actually this infection started within me, I can hit upon onto my childhood days. And the first mentor of my life is my dad. He's the first person who introduced me to science. Not only that, along with whatever a dad tells to his daughter, that I'm the most amazing daughter that anyone can have, he had a strong belief that I have a career in science. And that inspired me, motivated me to pick up research as a career, and I went ahead pursuing my doctoral study with Professor Jagat Roy, Cytogenetics Lab, BHU. Thereafter, I took off for my postdoctoral studies at UCLA, and here is where things started happening great. I got an opportunity to work in a project spearheaded by two incredible scientists of UCLA, Professor Volker Hartenstein and Professor Utpal Banerjee. Now, these two incredible scientists were pole apart in their attitude towards science. One was very poised, philosopher-like. He was artist, he used to paint, and he was a part of Los Angeles choir, so he used to sing. The other person, Utpal, was a go-getter, incredible scientist, go-getter, extremely ambitious, and he used to keep us on toes. This deadly combination actually was a great thing for me. I underwent a metamorphosis, and the whole trajectory of my career completely changed. They are the fellows who introduced me to the study of blood cell development using fly as a model. With Volker, I worked on the embryonic part. With Utpal, I pursued the larval part. And as an independent PI, I have introduced adult fly as a great model to study hematopoiesis in Mohali. Before going to the details of my work and my journey, let me introduce you to fly hematopoiesis per se. The three cell types that constitute the blood cell population in fly are very simple. The plasmatocytes are the predominant population, are professional macrophages involved in tissue remodeling and phagocytosis. Crystal cells, as the name implies, have crystalline inclusion and are involved in wound healing. Limerocytes are rarest of the kind and can only be seen in an infected individual. Though these three cell types are extremely simple in their structure and function, when you compare them with a vertebrate counterpart, what is amazing though is the conservation of the signaling molecules and the transcription factor that it takes to make mammalian blood have their ortholog doing the similar job in fly. During the early period of my postdoc, I was able to also demonstrate that there is a conservation at the level of the origin of blood cells across this divergent taxa. Like vertebrate, Drosophila hematopoiesis is biphasic. The first one is primitive hematopoiesis like ours, happens in the head region of the embryo, while the definitive hematopoiesis takes place in a specialized organ known as lymph gland. This gland develops in embryo, becomes functional in larvae. With the onset of pupation, it ruptures to release hemocytes into circulation. A little more onto this hematopoietic organ. It's a multilobate structure on either side of a fly heart or the dorsal vessel, which is skimmed up right here. Trust me, this is not just a bag of blood cells. Using molecular markers, we have been able to identify 3 distinct zones within this developing organ. The outer ring is populated by differentiated hemocyte, and we call it as cortical zone or CZ. The inner zone comprises of tightly packed cell rich in cadherin and sharing several characteristics of stem cells. We call it medullary zone or NZ. And right about here is a group of cells which are responsible for the maintenance of this stem-like precursor, and we call it niche. The beauty of the system is that we have tools and means by which we can manipulate each of the cell type and assay the effect of the manipulation onto the near neighbor as well as the distant neighbor. What have we learned in the process? During my postdoctoral journey, along with Utpal and Volkan, we have worked out multiple ways of progenitor maintenance. One is very simple, the instructive signal emanating from the niche responsible for the maintenance of the progenitor. We also discovered a signal that is intrinsically generated by the stem cell for its stemness, and a very interesting signal that is evoked by the differentiating cells impinges on the self-renewing progenitor and quietens it down. So there are three ways in which the progenitor maintains itself in flight. Now papers were happening. I was getting nicely trained by Utpal and Volkan, but I was very motivated to come back home. So the science that I'm learning up there, I wanted to translate it right here, set up a lab, have my own lineage of students, train them so well that they also perform equally like any student across the world. So having said that, I had actually applied in many places. The job that I finally settled down was a job offer from IISER Mohali, Indian Institute of Science, Education and Research. It was at its infancy. I decided to join the place. Well, it's a time in which I had to snap the umbilical cord, move out from my comfort zone in Utpal and Volka's place and dig out a life of my own in India. So I should have a survival brought in place, the recipe of which I thought would be a good helping of passion and motivation blended by lots of patience with a dollop of dreams and churn it out with realization. What kind of realization? Three is which I started off. First, that I'm moving into India, things will not go as fast, as smooth as it has been going in UCLA. Second, that I'm starting in an institute that is in its infancy, so I have to build up a lab right from scratch. And third, which was something was very important for me, that I was very well cocooned in a corner, doing my experiment, getting my papers. That was only because my PIs had shielded me off from the financial and administrative issues. And I admired them for that. But that is the point, was my learning experience. When I started off as an independent PI, I underwent a transformation. And that transformation was indeed needed for increasing the quotient of patience within me and the confidence that I can alone handle any issue. What are the kind of issues? I'm very happy that yesterday Sri Laja introduced few, that you have to talk to several levels of people. If you are building up a lab from scratch, from engineers to architects to mundane laborers to electrician, and the language is different for everyone. You have to well equip yourself. What are you actually aiming for? If they know that you are aiming for this thing, they would be able to help you out. If they realize that you are clueless, they would be overpowering. So it's better to know what you are actually aiming for. Apart from your dedications and your involvement in making up the lab, what else I did? Of course, I entered into the constant conversation with the purchase department, cracking deals with the vendors. I want this instrument and I can't pay 5 lakhs for it. And then I fought with them to get the guarantee extended from 2nd year to 5th year. Okay, so it's a plethora of things that you have to deal as an independent PI. Along with that, since it is IISER, teaching is a must, and I started enjoying teaching. I started mentoring undergraduate and graduate students and started taking some PhD students. So the last 5 years, I don't lament, I have slowed down a little bit, but it's a transformation, a well-needed transformation to become an independent PI. One thing I did, it might sound silly to someone, but it works for me all the time. Before leaving US, I made a document that listed out the reasons why I'm leaving for India, right? So whenever I got frustrated or irritated or worried, I just pull out this document and it actually reinforced my motivation. So this is a slice of land that was given to me in 2010 by IISER Mohali in the transit campus. The campus was extremely small, so I would have to do all my experiment along with my colleagues in the central lab, but I wanted a specialized Drosophila lab, so about 180 square feet area was taken out and given to me. So we started partitioning and building up the lab somewhere in 2010. Yes, so the first stereo binocular set, sponsored by my home institute, appeared sometimes in April 2010, and my students are very glad. So with the minimum, armed with the minimum infrastructure, binoculars and a fly lab, I felt like I'm very tempted. I have to start my work. But what kind of work I do at this point? I thought that I was armed with a good training in genetics and a good understanding of stem cell biology. So what can I do? At this moment, I thought that, okay, people all over the world, including my postdoctoral lab, they were interested in understanding how stem cells maintain themselves, how stem cells crosstalk with differentiated cells, how the stem cells talk with its own niche. But no one was caring enough to know what is the mechanistic basis of the niche maintenance, because that is more important to maintain the sanity of the stem cell. So I thought, well, that's an idea that I can hit upon, and I started writing down a proposal, submitted it to Wellcome Trust Intermediate Fellowship Competition, and well, not only my flies have wings, I also got my wings thanks to Wellcome Trust. You know, armed with this fellowship, it not only gave me a mental satisfaction, it actually endowed me with lots of power to build up the laboratory. Along with my colleague, Dr. Sudip Mandal, who happens to be my husband too, I set up institute-sponsored confocal facility right at 2010. Started off my project, Longevity project, which was aimed in doing a genome-wide RNA screen to find out modifiers or players important for niche maintenance. And I won't go in much details, but what we got was really rewarding. We got several genes whose function, when attenuated from the niche, affected the niche number and its functionality. On the other hand, we had some negative regulators. Why negative regulators? Attenuation of this transcript from the niche expanded the niche, and in most cases, the niche lost its vitality. Currently, we are concentrating on few of these molecules, few of these candidates, and we are carrying out detailed molecular analysis to find out the exact role that these candidates play in the maintenance of the niche. Meantime, our final campus was ready. This is how it looks if you arrive here at Aisar Mohali as of now today. So the road will take, all roads take to Aisar Mohali. This is the archway or the main gateway through which you can enter, right? So this is the picture taken, say, about 2 months ago. This is my laboratory space in Aisar Mohali campus, 2010, given it to me. Lolitika, this is your lab space. We are going to put together the floor and the wall. Rest is yours. Go plan it out. With the full institute support and with the continuous support from my colleagues as well as bunch of excited undergraduate and graduate students, I went ahead to build up the lab. The journey was little long, but the ultimate result is what I'm very happy about. I have a functional fry room and a dry lab in the main campus, which I moved in 2013. I have a wet lab that I share with Shudip's group, which is also well equipped. And then I managed and squeezed in a little space, about 280 square meters, wherein I built a cell culture, microinjection and laser dissection facility, which is up and on from 2014 of March, right? This has been a long journey. I joined somewhere in 2009, July, and I finally got a final lab done in 2013. But trust me, I was never frustrated. If you can allow me to use the language, I'll never mind about it. I thought this was my third pregnancy. It's a prolonged pregnancy. I knew something is happening with lots of expectation. It's my baby. It's taking time, but I have put every drop of it to create it. So I own it. And in the journey, the first batch of graduate students who were with me were so enthused that really I don't have to tell them go clean up the lab, ensure the lab things are in proper condition. They know it all because it is their lab. In the process, I know if there is a sudden power cut, I know which MCB has tripped. Go, finish it up, 10, 12, 15 must have tripped. I know the entire cycle. It's my lab, so it's a baby of mine. Third baby in the sense, I have two daughters, one in class 10 and another in class 5. Well, having butter project sponsored by Welcome DBT, rocking, I thought it's a time to take a challenge. A challenge which might land me up in mess, but never mind, I have a Welcome DBT project which is working fine too. So what did I ask? I asked a question which is a little bit strange, but let me walk you through. This slide I have already shown you few in the early part of the presentation, that active hematopoietic sites have been identified in embryo as well as in the larvae. So the head mesoderm and the larval organ, which is the lymph gland. People believe that the adult fly doesn't have any active hematopoietic site. This is a general belief of the field, including my postdoctoral lab also. What I argued is that compared to an adult, compared to a larvae, the adult faces diverse and complex challenges, and therefore leaving onto the contribution. So the adult, which was supposed to be believed to be leading on the contribution of these early stages, was a kind of a belief of the field, right? So we argued that the adult, which faces much more challenge compared to the earlier stages, it's really insane to believe that they can survive on the contribution of early stages, and therefore something is going on there which we are missing. So let's start a quest to find out the adult hematopoietic site. And when I actually asked my student who is going to volunteer, one fellow in his second year said, I'm all with you, and there was a master student said, I would do as a master project. So a master student and a graduate student and myself, we went ahead with this question. First we looked at the fly with several reporters, markers, and enhancer trap lines, and true, it was a chaos. It seemed that hemocyte, which is marked green here, is present all over the place. So there is no definite structure that might act as a site, right? But then we started careful dissection, fillet preparation of different time point of the adult life, and see what we get. So this is an adult fly abdomen. I'll take you through a movie. Slowly and surely you would appreciate. Along with the sporadic hemocyte present throughout the cuticle, a pattern emerges. A pattern emerges on the either side of the dorsal midline, that there are cluster of hemocyte present in each abdominal segment. And trust me, once we located this kept on appearing. And we went in a spree of dissection, 50, 100 flies. Every time on careful dissection, this structure popped out at us. We wanted to know more about it. And therefore, more about it, position. Where is it actually located? So this is again an adult fillet, larval fillet. This is the anterior side, this is the posterior side. What we have done is we have used phalloidin, which marks the dorsal vessel or the heart proper of the fly, and the body wall muscle in each segment. The movie is taken from the ventral side to the dorsal side. I will roll it down and you would appreciate as we move towards the dorsal side. As we move towards the dorsal side, the hemocyte, which are marked in green, starts appearing. So one thing is clear, they are dorsally situated. And another thing becomes pretty clear, that they are situated on top of the heart. Is it that simple? We wanted to know a little bit more to this. So we made a transverse section in the confocal stack, and let's see what we see here. We see this is the heart lumen, which is a holy hole right about here, surrounded by muscles. These are the longitudinal heart muscle. This is the transverse heart muscle, which actually makes this lumen. And these are the body wall muscle that I showed you present in each segment. So there is a group that is created which actually nourishes or houses these hemocytes, which are marked green. So within this group created by these muscular entities, the cuticle actually flanks from the top. So the hemocytes are actually situated in a secluded group away from the entire body cavity, flanked by the dorsal cuticle and supported by the body musculature. Fine, they are not just sitting. What we realized that the hemocytes are fenestrated in an intense network of ECM. And what was rewarding that, the ECM molecule, pericardin, which was in close association, as you can see here, the strands of pericardin wrapped around the hemocytes, which are green in color, Pericardium is actually a homolog of collagen 4-like, which is known to be enriched in bone marrow, right? So we were excited. We wanted to know, is this association is of any significance or not? What to do? Well, it's fly, and let's use genetics. So we use a mutant, which is a loss of function for the receptor of pericardium, and its name is Lolitika. In a Lolitika mutant, as you can see, pericardium expression compared to adult is highly compromised. And what is very remarkable is that the repetitive fashion of this cluster, which is otherwise seen in normal fly, is highly compromised in a LOH mutant. That itself signifies that the ECM significance is there in maintaining this cluster. We were more excited when we found that the other component of bone marrow, laminin A, is also expressed here. And on downregulation of laminin A from the cluster, we actually could see that the cluster fails to form. So therefore, we have demonstrated that the ECM is in adhesive interaction with the hemocyte and is an essential entity to build up the hub. Now, why hub? I'll take you through. Till about now, I've shown you the clusters are situated at the dorsal side of the heart below the cuticle. Hemocytes are located at a muscular group created by heart muscle and bodyguard muscle. Hemocytes are tightly associated by the ECM network, which is critical for its maintenance. This anatomical location is separated from rest of the abdominal cavity by dorsal and pericardial diaphragm. We wanted to know what is actually present in the heart. People say that the adult survives on the contribution of embryonic and larval lineages. Is it true in this case? Yes, that is true. We could use embryonic marker and larval lineage study. Could see that yes, both of them are quite present in this cluster. That's not interesting. But the whole exercise was interesting because we found the progenitor. That was not reported till date. Using a cocktail of markers that will label the differentiating cell and the progenitor, we ended up locating these red cells, which are the progenitor cells, within this cluster. Due to shortage of time, I couldn't show you, but we were able to show in vivo specification of this progenitor into crystal cell and plasmatocytes. I think I have a slide to show you about plasmatocyte. So using, Drosophila genetics is it's a boon. It's a boon. Whatever question you ask, whatever problem you have, you can find an answer using Drosophila genetics. So we use a lineage tracing cassette that will not only enable us to lineage the hemocyte, but will also birth date the hemocyte. So in a nutshell, if you see a green cell that was born a long time ago, something which is red is just born right now at the time of observation. And if you follow the arrow, I have marked out the cells which are red but not green. So we could show that hemocytes are being born or differentiated within this hub. So this is something which was never reported previously. We could demonstrate that no, the adult is not solely dependent on larval stages. It has its own hematopoietic cycles. Where do the progenitors come from? Well, I walked back in development, and this is the organ that I had shown you previously. It's a larval lymph gland. The primary lobe houses the differentiated hemocyte, while the secondary and tertiary lobe are known to house progenitor cell. So therefore, both student and myself, we hypothesize maybe the progenitors are coming from the secondary and tertiary lobe. Is it true? We have to prove it, right? So we created a situation, a genetic situation, where we ectopically induced differentiation in the reserve population, right? And we ended up getting a genotype where secondary lobe is differentiated along with the primary lobe, but the tertiary lobe and the quaternary lobe are shielded or buffered up from differentiation. That being said, that means they still house the progenitor. So my student said, let's now look at the hub of this genotype. What do we see there? This is the wild type hub where you see the green cells are plasmatocyte and red being the progenitor. When you use this genotype PVR, which is known for cell migration, you landed up showing that there are lots of progenitor. And the crux is you have actually created loss of function of PVR only in the plasmatocyte population and not in the progenitor cell. So we kind of started believing that the progenitor cells that are derived from the secondary and tertiary lobe are the ones that are seeded off in the hub. But then we have to prove it again. The way to go is to initiate a screen to find out new markers that are restricted only to the secondary and tertiary lobe. And then we would lineage trace this lobe and ask what is the fate of this lineage trace cells in an adult population. And we could very clearly show that these lineage trace cells, which are green in color, either they lack any of the differentiating markers, or there are cases in which they are positive for both of them. So we are now very sure that the progenitor cells are coming from the reserve population and they are bipotent in nature. They can give rise to either plasmatocyte or a crystal cell. We then ask that the plasmatocytes sitting in the hub, are they efficient enough to mount response to any insult? How do we go? We challenge the fly with RFP labeled bacteria, and as you can see here, this is a high mag image of a hemocyte into which few bacteria are trapped. So we have shown that the plasmatocyte within the hub are primed and ready to mount response to any insult during adult life. Not only that, we could demonstrate also that the hemocytes, which were believed to be locked in senescence, can be induced to re-enter into cell proliferation upon infection. This understanding brought a paradigm shift in fly hematopoiesis, because now we can use adult fly as a model. Till about now, whatever studies we have done was restricted to the immature stages, embryonic and larval stages. There was no report of adult hematopoiesis going on, so that is a contribution towards basic science. But you will be amazed what else we can extrapolate from this observation, which due to shortage of time, I couldn't show you. We have demonstrated that the ECM, laminin A and type 4 procollagen, which are present in the bone marrow, is also present in adult hematopoietic hub. We have clearly demonstrated that cells migrate to and fro. From the bone marrow, it demonstrated, it was known, and we have shown it in the adult hub. We have shown that the adult hub is a site of new blood cell formation in adult life. We have also demonstrated the presence of heterogeneous progenitor cells. We have also shown that non-signaling, which I couldn't show due to the scarcity of time, involved in lineage decision. We have sufficiently demonstrated by genetics that there is a homing of progenitor cells within this adult hub, just like bone marrow. We propose this is a site of extended definitive hematopoiesis like bone marrow. We have also demonstrated that the cells within this hub are so slow cycling and are capable to respond immune challenge. The entire work is in press now with developmental cell and would be online on 26th of May. So, all being said, this is my group at Tyson Mohali. Shirkat Ghosh is a student who did bulk of the work, and master student Arshdeep Singh did the peripheral job. This is my entire troop. I have a battalion of undergraduate students working with me. I am really in depth with this person, Suresh Kumar, who actually supplies endless food and wash bottle. My collaborator is Dr. Shudeep. And this is again a snapshot of Tyson Mohali. The hospital is attached at the back. It's just when you come out from your laboratory. And in debt to Wellcome Trust DBT Indian Alliance for its continuous and support, as well as intellectual support too. My institute, which has been great in helping me to set up the laboratory, my director, my head of the department, colleagues, it is a journey to remember. And CSI fellowship to Shirkat, Department of Biological Science, it led to the fly community, Hybridoma Bank, Bloomington Stock Center for the stocks, and of course to you all.